I'm going to talk a little bit about some of the work we've been involved in over the last 18 months or so. And first, I'll just tell you a little bit about my view of systems immunology, because it's kind of a buzzword that people throw around. But for me, as an immunologist, the way I think about it is that it's kind of a shift in focus from traditional immunological investigation, which is largely focused on individual white blood cell types and their function and regulation, to a focus more on how different cell types interact and collectively give rise to various forms of immunopathology or productive immune responses. And so when I talk about systems immunology, that's what I mean, understanding these interdependencies. And there's a lot of technological breakthroughs in the last decade that has enabled this sort of investigation now, involving single cell methods for cellular analysis, obviously both proteins and mRNA and genes. And then also plasma proteins, which are now readily available to measure at scale of hundreds of proteins from a small blood sample. And obviously all the transcripts and DNA sequences and so on. So the way that we typically go about addressing a number of conditions in human patients is by using these systems level analysis methods in combination with longitudinal sampling. And the reason for this is because each and every one of us has a very unique makeup of immune cells and proteins in our bloodstream. These are very variable between individuals, but quite stable within an individual over time in the absence of immune activation or stimulation. But by monitoring an individual over time, we can see changes such as this cytokine increasing, and right after that, a activation of a particular white blood cell. And by using clever informatics in combination with these longitudinal data sets, I think we are able to actually uncover some of these interdependencies between cells and relationships between features in the immune system, even when humans are incredibly variable. Another thing that I just want to highlight, because it's been really kind of transformative for our work, is to be able to think about ways of overcoming this massive variation among humans. And one way to do that is to minimize the technical sources of variation, because we really do introduce quite a lot of technical variation by the way we process samples. And so we've developed methods to preserve whole blood directly at blood draw, and then do downstream cytometry analyses. We're currently working on mRNA analyses. And by doing this, we really reduce the amount of variability. First of all, we capture all the white blood cells, including the neutrophils, which are lost with PBMC, which is the normal sort of standard cell that people use for these kinds of studies. But also, and more importantly, I think, is if we look at the technical replicates here, the coefficients of variance, so the variability among technical replicates, is always much lower in preserved whole blood than in PBMC. And the reason for this is because PBMC preparation is never done the same. It's always, slightly variable in temperature, in time, and, location of the moon or whatever. And so all of these things introduces technical source of variation. So that really prevents us from seeing the biological patterns we really care about, unless we have massive cohorts, which we rarely do. So anyway, this was just kind of a intro to the way we think about these sorts of studies. And then currently during the pandemic, we've dived into these different disease manifestations induced by the same virus, which is the mild versus severe COVID-19, MIS-C. As a pediatrician, I've treated about 100 patients with MIS-C, and so this has become a really important topic for us to try to understand. And then currently we're looking a lot into persistent symptoms, especially severe long COVID symptoms in previously healthy individuals. And we work within the context of this really large global network, which was initiated by Jean-Laurent Casanovas at the Rockefeller, as well as Helen Su at the NIH mainly. But it involves colleagues all over the world. And this network has so far, it was inspired by the idea that rare presentations such as severe COVID-19 in previously healthy young individuals might uncover a previously unrecognized immunodeficiency. And in contrast to that, unexpected resistance might be explained by either immunological features of resistance or very potent immune responses, let's say in the elderly that do not develop severe disease, for example. And so in this context, two studies have been produced so far with a number of collaborators, as you can see, and we've contributed a small part to these. And what these have basically explained is a fraction of patients under 50 years of age who have developed very severe COVID-19 pneumonia. And about 4% of them have been explained by inborn errors of immunity, either in the viral sensors or in signaling mechanisms downstream of the sensors. But a larger group, about 10% of the individuals, have been explained by neutralizing antibodies to type 1 interferons, which prevent the host from really responding properly, despite otherwise having an intact immune system. And these sorts of autoantibodies have been well known in other diseases, such as APEX syndrome, which is air deficiency and autoantibody disease, and also in lupus patients and so on. But it was not known that they were so commonly seen also in previously healthy individuals. And so the common theme that has sort of come out of this, and I'm inspired by some discussions with Akiko Kawasaki at Yale here, but basically initial early robust interferon responses limits the viral load and typically leads to a milder disease. And this is what we normally see in young people and sort of most children especially. And then in the elderly, there's a delayed interferon response, partially due to the virus, which inhibits the interferon response, but also perhaps related to viral load and so on. These things lead to a more severe disease and more of an inflammatory response. And in the absence of an interferon response due to immunodeficiency or autoantibodies, the viral load goes through the roof, and these patients always develop life-threatening illness. And based on all this logic, current trials are ongoing to provide interferon at positive testing in early phase of disease to try to mitigate this disease. So this is sort of where we are in my understanding of the field in COVID-19. I'll skip this. And what we tried to contribute early during the spring of 2020 was an application of our sort of systems level methods of analysis to patients with severe COVID-19. And we teamed up with colleagues in Finland who had a very sort of rigorous sampling mechanism, sampling patients frequently in the ward and then ICU. And I wanted to show a couple of these things that were a little bit surprising to us. So for example, here, what I'm showing you here is single cell mass cytometry data showing eosinophils only from one patient as an illustrative example. At the time of admission to the hospital here, the patient was stable but required oxygen. At day 2, there was an expansion of an eosinophil population in the blood expressing the CD62 ligand. And right around this same time point, day 4 or 5, these patients develop ARDS. And we see also a slight increase in gamma interferon. These cells have been reported in other conditions, such as severe asthma, to respond to gamma interferon, go into the lung, and then contribute to pathology in the lung, inflammatory responses. And so we think these eosinophils might be of interest to study further. We see similar patterns for a type of monocyte population that express the fractalkine receptor, a receptor for a chemokine, fractalkine, involved in cells that follow along the endothelium and then leave the vasculature. And so we think this is another important pathway to keep an eye on when understanding severe COVID-19. And as you all know, there's a number of mediators that have been shown to be important in the lung during the severe pneumonia. We argued together with some colleagues here in Sweden that maybe it's difficult to sort of target one of the cytokines at the time of this ARDS or severe pneumonia, because there's so many mediators involved. And I think the trials are showing that even though IL-6 blockade has shown some benefit, the real benefit is seen with preventive sort of steroids prior to the pneumonia being this full-blown hyperinflammatory syndrome. And so we have teamed up with a Swedish company called Cancera, who have a inhibitor to this particular fractalkine receptor. And the logic behind this is that instead of waiting for the cells to leave the bloodstream and go into the inflamed lung and try to prevent the inflammation there, how about preventing them from leaving the blood vessels and going into the lung in the first place? And so we are currently conducting a randomized controlled trial in Sweden. It's just concluded because we don't have any patients left to recruit nowadays, but we hope we'll be able to analyze some data and give you some feedback soon about whether this worked or not. But basically the idea is to block the fractalkine receptor early in the disease course to try to prevent severe pneumonia in COVID-19. And as I said, we hope to be able to report that soon. We have looked at, in this cohort, also at the determinants of the antibody titers. So what are the determinants that sort of tell you whether an individual is going to respond with high titer antibodies or not? And we see that strong inflammatory signals such as gamma interferon in the blood, high IL-6 levels, those are negatively associated with titers against RBD, while basophil levels are positively associated with that. So this is in line with some historic data looking at flu, for example, where basophil seems to have an impact on B-cell responses. So we thought that was quite intriguing and also something to look into a little bit more maybe. I'll skip that for sake of time. But then getting into this condition of MIS-C then is, you know, in the spring of 2020, colleagues around Europe and then also in the US, especially in New York, reported high incidence of this, of patients with Kawasaki-like disease, which didn't really fit the pattern of classical Kawasaki. These were mostly older kids. They had a lot of abdominal symptoms and severe myocarditis. And so we, together with other colleagues in Italy and elsewhere, decided to start studying these patients using our sort of systems level analysis for immune profiling. This is data from New York, actually, and just to highlight this delayed incidence following the peak of positive tests in the New York area, spring of 2020, followed one month later is the sort of incidence of MIS-C here. And why this delay is a subject of some debate at the moment, but it's pretty clear that that's the case. And so this is what we did. We had a historic cohort of Kawasaki disease children sampled in 2017 and 18. So we know that this disease is not triggered by SARS-CoV-2. We compare that to some healthy infants and mild SARS-CoV-2 infection, children with such mild infection found usually through screening, many of them asymptomatic. And when we look at their immune profile, so we're starting off here just showing cytokine profiles. So this is principal component analysis of 112 unique plasma proteins. What you see right away is that adults with hyperinflammatory acute COVID-19, so these are either in the ICU or in the floor being treated for acute COVID, they have a very different cytokine profile from the MIS-C patients. And these also differ from the Kawasaki disease patients. So there are 3 gray dots here, which I think are misdiagnosed patients with probably having Kawasaki rather than MIS-C in this case. But they are clearly discrete groups, and there are specific cytokines that differ here, which I think is important to keep in mind when when understanding MIS-C pathology. Some of these distinguishing features is Kawasaki is very much IL-17 driven pathology. This has been shown by others, showing Th17 cells in the blood vessels of coronary artery biopsies, for example. Kawasaki disease patients also have really high IL-6 levels, and MIS-C is milder in this respect, more similar to patients with mild SARS-CoV-2 infection. There are biomarkers of vasculature, vascular damage or endothelial inflammation in both of these conditions, such as this one and some of these other ones, and suggesting that there is a vasculitis sort of component to MIS-C, which is not surprising given the clinical presentation. The way we treat these kids normally is by steroids and immunoglobulins, but we have also been quite aggressive in Sweden with IL-1 receptor antagonists, anakinra. And I think we are currently looking at the data, but it seems like we have a much lower incidence of ICU admission than most other countries, perhaps because of this very aggressive immunomodulatory therapy up front. So what is causing this pathology? Well, we turn to the study of autoantibodies, given the fact that in Kawasaki disease, it's been reported that autoantibodies target blood vessel structural proteins. And so we performed arrays of autoantibody binding against 9,000 human proteins. And we found a number of these. So, and many of these are proteins targeting structural proteins, such as in the heart, for example, blood vessels and so on. There were some that were quite different though between Kawasaki and MIS-C, such as these casein kinase proteins here. And this became a little bit more intriguing given the fact that Nevan Krogan's lab at the UCSF reported in a study that cells infected with SARS-CoV-2 show a marked upregulation of specifically these casein kinase proteins. And if they are inhibited using a chemical pharmacological inhibitor, the virus does not replicate. And so this seems to, the cell seems to be important. This protein seems to be important for the virus to replicate in cells. Anyway, another theory that has been proposed specifically by Moshe Arditi's lab at UCLA is that T-cell activation in MIS-C might be triggered by a spike protein superantigen motif, which triggers cross-linking of specific beta chains of the T-cell receptor, specifically the V-beta 11-2 here. And they have reported, and following them, 2 additional studies have reported expansion of this V-beta 11 in patients with MIS-C. So perhaps that is one clue. It's still a little bit unclear as to why the disease would be delayed with 1 month in the case of a superantigenic stimulation, but that remains to be seen, I suppose. So I have to thank a number of people here in my own team, colleagues, former and current, as well as collaborators. And I am happy to take questions if you may have any. Let's see if I can stop sharing.
All right. Thank you very much. Beautiful, beautiful talk.
Thank you.
At the beginning, let me start at the beginning. You mentioned the issue that people who are low in interferon, either because of autoantibodies to interferon, do badly.
Right.
And so the thought was to treat with interferon early.
Right.
But don't you have to worry that you'll make things worse, especially, I guess, if you treat too late?
Yeah, if you treat too late, I think that's probably a bad. Oh well, you won't alter the course of disease probably because you want to limit the viral replication early on. I mean, these trials, and that's obviously the challenge, right? How do you find a patient to know that they will be in need of interferon supplementation early on? Wait, Sola, I have a guest here, as you can see. So I think those trials are ongoing, and we'll see what they give, you know. And so far it seems pretty promising, especially maybe the lambda interferon, which would be the local mucosal interferon. So we'll see what they give.
All right. Let me see. I don't have any in my box.
Peter, this is Eric. I have a simple, perhaps naive question. I'm wondering if you could comment on the longer term prognosis of the kids who are coming up with MIS-C, and are some of these issues resolving themselves as time progresses?
Yeah, so it's really interesting, actually. The very severe patients clearly need treatment, or they'll die because they have, a malfunctioning myocardium, and their blood pressure is low, and their encephalitis, and all these other really serious, problems. But we also do see that there is a category of MIS-C patients that are either missed, never diagnosed, and that do seem to recover spontaneously. In my experience, from about 250 patients here in Sweden that I've one way or another been involved in, we don't see very bad sort of long-term consequences. They've all sort of recovered after treatment. We haven't had any deaths. And even though there's some mild neurocognitive sort of problems later on, nothing really severe other than that. Which is really fortunate. We've been very worried about many of